wanting you to think it's an assassin bug, but it's a leaf.
Actually,
I can't decipher probably.
but when I was doing 3 lab days a week,
It looks like a Regidean with the...
It does kind of, and I'm not 100% sure with that. I think it's a will.
Now I'm doubting all of your...
50%
Yeah, I
like,
know. It's all a lie.
All right, we're going to get started today. So today we have a very special guest, Dr. Will Kuhn from Discover Life in America. Will has kind of a unique job where he's the, what's your job title? Director of Science and Research for an organization that's trying to document all the diversity in our most diverse national park in the United States. Will did his master's work at Virginia Tech with Dr. Rod Youngman, and then moved to Rutgers Newark, where he did his PhD with Dr. Jessica Ware. And then he received an NSF postdoctoral fellowship to digitize dragonfly species for identification. So took a bit of a switch moving to Tennessee, but he's a really good guide if you get down there and want to look at some of the cool species that are down there, right?
Mm-hmm.
All right, thanks, Will.
Thanks, Ann. Is this doing anything? Can you all hear me in the back?
no, it's not on.
It was.
If you turn the little battery pack on, you can turn it on.
This is my first time using them.
It was scratching. Is that
It's scratching.
Doing? Okay.
Can you guys hear now? Okay, I think I just didn't have it on high enough.
Sorry, everyone online. I'll just do this. Ooh, I love the insect Legos over here. Okay, digression. So, hi, I'm Will, and as Ann said, I work for Discover Life in America. And today I want to talk about biodiversity and how we are looking for biodiversity in the Smokies. Maybe. So, Discover Life in America is one of 4 nonprofit partners of Great Smoky Mountains National Park, all serving different roles to help the park along. There's Great Smoky Mountains Institute at Tremont that does a lot of in-house education. There's Smoky's Life, which actually just rebranded, used to be Great Smoky Mountains Association, and they run all the bookstores in the park, but also we work with them to create publications. They create all these super cool Smokies specific field guides, and lots of other things. And then Friends of the Smokies that raises tons of money to go back into park projects. Some of that money comes to us. So our mission as a nonprofit organization is connecting communities and scientists in discovering, understanding, and conserving the natural world. So we have a very biodiversity-focused mission, and I'll tell you a little bit more about the history of that. And we are focused in Great Smoky Mountains National Park, which is really big. It spans the state line from Tennessee to North Carolina, and it's over 500,000 acres in size, spanning 5 counties, 3 in Tennessee, 2 in North Carolina, and it's the 4th largest NPS unit in the eastern U.S. It's about a day's travel from some ungodly percentage of the U.S. population, and so it's also the most visited park in the country.
Ungodly as in good.
Yes, good. Yeah. So a lot of the population on the eastern U.S. is around there. So we have 13. I think we had 13 million visitors last year. 14 was the record set a couple of years ago. It's a lot of people, but also a lot of biodiversity. Some of the things that make the Smokies such a biodiverse place is that the mountains are very, very old. They are much older than the Himalayas or than the Rockies, and they actually used to be taller than the Himalayas and have weathered down over millions and millions and millions of years. And a lot of what used to be the Appalachian Mountains and the Smokies is now the sand that's on the Florida coast. and that has been fed down the Mississippi. So a lot of the Gulf Coast sand is used to be mountains. It also has this really interesting complex topology. It's some of the higher, I think it is the highest mountains in the, or some of the highest mountains in this Appalachian mountain range that expands. You have a little piece of it in northern New Jersey, but yeah, the topology or the highest mountain there is 6,643 feet, Clingman's Dome, and then the lowest point is Abrams Creek out on the western side of the park at just under 1,000 feet. So there's this huge range of elevation, and lots of kind of rugged mountains that make for tons of different niche space. It also has tons of water. So there are 2,900 miles of streams, most of which, except for one, originate in the park and flow out of the park. It receives in places upwards of 8 feet of precipitation a year, so it is a temperate rainforest. That's a lot of precipitation. It has a ton of all this rain fuels plant productivity. So there are a ton of different vegetation communities, depending on how you count them. Somewhere between 11 and 79 different vegetation communities, just all across the park. Each one of them harbors just a ton of biodiversity that goes along with this plant community. And so it is probably the most biologically diverse U.S. national park in the country. And yeah, for all these reasons and more, and it's just a beautiful place to work. But there are some threats to this biodiversity. One of them is development. There are a ton of, I've heard a couple of people say they've been to Gatlinburg. Gatlinburg is one of several kind of gateway communities to the park. And it is heavily developed. It's like a tourist trap. And there's some like encroachment on the park. You can see the park line very clearly because it's like hotels and tchotchke stores and then nature on the other side of the line. So development is a big thing. Air pollution is actually slightly less of a problem than it was a decade or two ago because several coal-fired power plants in the area have been shut down. But there's still some air pollution problems. Climate change is an especially upcoming problem, considering there are a lot of high elevation habitats that are going to be slowly moving up until there's no up to go to. There are a lot of extreme weather events. There were a couple of different tropical storms that passed over the park last year. We had a big fire in 2016 and crazy wind events pretty frequently. We have a bunch of different invasive species, like the hemlock woolly adelgid has wreaked havoc on the eastern hemlock trees across the park. A whole list of invasive insects and plant species that the park is constantly having to keep an eye on and try to mitigate the effects of. And as I mentioned, there's a ton of visitation. So over-visitation is in itself a problem, and the park is constantly having to balance the need for having a good visitor experience, encouraging people to enjoy nature while also conserving the natural features of this landscape. And so our mantra as an organization is that we can't protect a species if we don't know it exists. We have to know the park has to know what species live here in order to be able to protect them. And so back in 1998, we were formed as an organization to manage a project that was first posited by Dan Jansen and this is the project called the All Taxa Biodiversity Inventory, which is aimed at cataloging every single species that lives in this place, and to not just know that it exists, but to learn more about it. Specifically, we have kind of 4 key questions that we generally seek to answer. What species actually live in the Smokies? Most obvious, but also surprisingly difficult to answer after 26 years of doing this. Where do these species actually live, and when are they active? How common or rare are they? Are they endemic to the Smokies or the Southern Appalachians? We have a number of endemic species in the area. And then most kind of complicated to answer, but very important, is how do these species actually interact with each other? They form this network of species interactions, and if we were to lose one from one of these threats, how will that affect the network of other species that they interact with? And so currently the major facets of the ATBI in the Smokies are that we as an organization conduct our own biodiversity research. Most of that is kind of targeted to specific groups that we know are undersampled, and a lot of those groups are arthropods. And I'll talk a little bit more about that in a second. But back in the day, like in the early 2000s, there was a kind of a blanket approach to catalog just everything, sample everything. There were 2 years of just blanket malaise trapping that just collected untold lots of material. We still have a backlog of roughly 500,000 samples that we are working to go through from the early days there. But we also work with researchers, so there are a number of different researchers, something like 1,000 different researchers from all around the world who have done work in the park over the years, and a handful of those have been permits from Rutgers, but we could get some more. We also work with the National Park Service, so we have our office in the park, and work daily with several of the staff from the park. They make their staff available and make their resources available to us, and so it works out nicely. We also do kind of special projects for the park when they have a particular question about the biodiversity in some area, want to do a survey or something like that. And then we also work with community scientists. So kind of from the very beginning, the ATBI was founded on this idea of bringing volunteers and bringing the community in to learn more about the biodiversity and to be part of the discovery process. We also work to educate the public about biodiversity and to train the future of nature advocacy. So we have a summer internship program that I'll talk a little bit about at the end. This year we are hiring 4 interns, and they will roam the park with us, doing biodiversity surveys, learning DNA barcoding, and lots more. We have a podcast with 2 episodes so far, but it's a upcoming thing. One on fireflies and one on kind of the history of the ATBI. We have a middle school program for middle schools in our region that is kind of like a mini ATBI in their schoolyard using the iNaturalist app. And we regularly do bio blitzes with community members, and I'll show you some examples of that, and have a program called Smoky's Most Wanted, where we try to get park visitors to use iNaturalist in the park. So in 26 years, we've more than doubled the number of species known to the Smokies. Back in the day, 1998-ish, there were around 9,700 species known to the park. But now there are almost 22,000. And so 11,000 of those are new park records, meaning that they are species that were known outside the park but hadn't yet been documented in the park. But then 1,079 of those are new to science, meaning that they were described from material collected in the park, which is really cool. But we estimate that there are 60 to 80,000 species in the park, so we still have a long ways to go. And we're also working on a way of honing that 60 to 80,000 down a little bit using statistical analysis of our previous our data from 26 years of study.
Excuse me, I just,
Yeah.
maybe I missed it, but what groups are you including? I mean, I'm assuming you're not doing bacteria.
Oh, love that.
Bacteria?
Bacteria are included, although bacteria are very undersampled. Most of it is macro life. Yes, good question. Yeah, there is. I'll talk a little bit about some. Well, there's a study looking at bacteria, fungi, and nematodes from soil samples, and we've had a few kind of bacterial surveys and other small things, surveys, viruses, and such. But most of those are kind of
Localized?
localized. Yes, thank you. Yeah, so just to give you an idea of how many new things we document every year in the Smokies, this really varies from year to year, and usually has to do with like a researcher will come out with a paper that describes a bunch of new nematode species or something like that, and so we'll get a big uptick in species for that year. But generally the average is around 300 to 400 species a year. Last year we added almost 300 species, 298, and most of those were new park records, but a couple of those were new species to science. There was a couple of new salamanders. One salamander genus was subgroups has been split a few times recently, and there was something else. I'll think of it in a minute. Anyway, several new species to science. Last year, this is just gives you a general idea of what we found. A lot of the new species records were wasps, especially tiny parasitic wasps, that we think are just very, very undersampled in the park. There's just a ton of parasitoid biodiversity, and so we'll be finding lots more of those for years to come. I do want to point out a couple of new records, mayfly and two caddisflies, and one of the interns working in the park actually found that, and that would be Liz back there, one of your undergrads in the department.
What?
You were part of the discovery. You didn't.
Surprise.
Y'all-
I was just typing. I'm like, why did nobody tell me about the mayflies? I was working with people. What?
Becky said y'all found two new caddisfly species and a new mayfly as you were doing samples.
That sounds like a Caleb thing, but thank you.
Yeah, so Liz was an intern with the park's entomologist last summer. So it was super cool to have them. Yeah, lots of you can see most of the species that have been added are insects, although there are a handful of microbes. I don't remember what specifically microbes, but also some new. I think that was maybe one of the new science was a new lichen. We have a ton of lichen diversity in the park. So just to give you a general sense of how biodiversity breaks down by number of species, insects unsurprisingly make up almost half of the biodiversity there. Everywhere, yes, yeah. Which, yes, these trends follow generally the worldwide trends. We think fungi, especially non-lichenized fungi, are probably grossly undersampled in the park, and there's a couple of people working to do more surveys there. We have a ton of vascular and non-vascular plant diversity. There are more native more native tree species in Great Smoky Mountains National Park than all of mainland Europe combined, which is just a fun fact. It's between 100 and 135, depending on how you define trees and native and such. And we have almost 1,000 species of lichenized fungi, lichens, and I always love to throw some shade on the vertebrates that bring so many people to the Smokies. We are the salamander capital of the world, 30-something species of salamanders. We have bears and elk and 250-ish bird species. And all of that fits into that tiny little sliver that makes up about 2.5% of the park's biodiversity. Extremely important, but not that biodiversity. Looking just at the arthropod biodiversity, this also follows general worldwide trends. We have a ton of beetle diversity, and beetles have been surprisingly well studied in the park and cataloged. But there are a ton of flies waiting in our backlog material, waiting to be identified, and probably many more species to be found. Again, probably lots of Hymenoptera that are waiting to be cataloged that haven't been cataloged yet. Lepidoptera have been, especially the butterflies, not surprisingly, have been reasonably well sampled, but we are still finding a lot of micromoth species, especially things like leaf miners. Hemiptera, probably there's some room for discovery. A lot of the aquatic insects have been reasonably well sampled by folks like Liz back there. But yeah, tons of mite diversity. And to give you a sense of which groups we found the most new things. So in each of these, we've got the orange is new species of science, the blue is new species records, and then the gray is like before we started the ATVI in 1998. So you can see beetles, we've added just a ton of different species, and quite a few of those are new to science. Same with flies and Lepidoptera. All the major groups, these also basically follow the normal trends. Oh, a ton of different mite species. Like most of the mite species in the park have been added since 1998. And in terms of percentage of species that have been added, not much in the way of, we only have 1 or 2 species of Dermaptera and mantises, but things like Symphyla or Strepsiptera, we only have 4 species of Strepsiptera that have all recently been added. But yeah, there are some groups that we have just added since 1998, and others that we have added most of our species since then. The harvestmen, there were just a handful of records before the ATVI. I'll give you a second to look at this.
Really, Protura and Diptura, you're like, yay, new species to science.
Yeah, yeah, tons of, yeah, same with Symphyla. I don't remember the actual diversity. I should have put the actual numbers on the right there, but still a lot. So currently, and the text here is tiny, you can visit this on our website. Also, all of these slides, I'll have a QR code at the end where you can go back and look at them and read this a little bit better. But just to give you a sense of how we're doing things now, we have we've been kind of prioritizing our activities. So we work with the park science staff to sort of continually prioritize things like undersampled insect groups. The bacteria, archaea, and protozoa are all high on this list. There are, yeah, we still think there are quite a few more, like mites and pseudoscorpions have been decently studied. what else? Anyway, so this is just an idea of how we're sort of prioritizing. Instead of kind of the blanket approach, we're refining and sort of targeting these different groups. And I talked about this backlog. So in what until recently was deemed the scary room or the scary lab, they were just bags and bags of samples and alcohol, most of them from these Malays trappings in the early 2000s. And so one of the projects we did the last few years was to go through and just like reorganize all this to at least get an estimate of the inventory of arthropods here, and they're mostly arthropods, and there's 500,000 of them. They're all in 70% ethanol, about 8,000 vials, and they were mostly collected from these 11 ATBI plots across the park. So in the beginning, there was an effort to kind of parse the park into different sort of types of habitat and to sample across as many of those different varieties of habitats as they could. And so they chose these 11 spots and sampled from them continuously for 2 years. Malays traps running, being checked every 2 weeks, and so we just have so many things left from that. And more than half of those are flies, plus a lot of wasps and true bugs are the highest represented groups there. So we have a lot of work to do there, and some of the ways that we are working through this, one of them is that we have a mini grant program. These are small grants that we give out mostly these days for people to identify material from the backlog or to identify material that we've already collected. We do occasionally give them out for, we think we have web spinners. There's a very good possibility that we have imbiodines in the park, but we have never found them yet. So we were going to, there's a mini grant in the works to pay somebody that has done surveys for web spinners elsewhere to come in the park and look for them. Things like that. So very like targeted, taxon targeted projects. And again, you'll be able to access this later. Keep moving on. Another thing is that we are using DNA barcoding. This is something we've recently delved into, and I've heard a lot from your students that there's a lot of DNA barcoding going on here as well. But this is a great tool that we can use for identifying unidentified things, but also filling in the genetic library for species that occur in the Smokies. Species that have been expert identified, specimens that have been expert identified, to go through our catalog and barcode individual representatives from those. And this is also a great teaching tool. So we've been working with the Citizen DNA Barcode Network at Cold Spring Harbor Lab in Long Island, and they provide all the materials and equipment to do barcoding for us to do in-house barcoding on a limited basis that we can use as a teaching tool. And the only caveat is that we have to teach other people how to use it. So we include it in our internship program, and we do workshops periodically, and it's been really fun, and we've gotten some new records that way. So as I said, we've been working with the Cold Spring Harbor Lab, but we also can send off specimens to the Canadian Center for DNA Barcoding at University of Guelph. And we do have to pay for samples, but it's not that expensive. Comes out to about $9 per sample, or $14 if they're degraded. They have a process for older specimens. And it works great. Everything's just uploaded to Bold, and so that's been nice for kind of en masse barcoding, whereas we've used the CDBN in-house stuff for kind of smaller scale barcoding. and I'm happy to talk more about either of these if anyone is interested. So as I mentioned, we are trying to get park visitors to use the iNaturalist app in the Smokies. So if you haven't used iNaturalist before, it's a super cool platform. It's an app, an online interface. There's another app by the same people called Seek, and these are all just really great. It's like a social network kind of thing meets biodiversity science. So you can upload pictures of an organism, or even sound recordings of an organism, bird calls and such, and other people will help you identify them. And it also has a machine learning, computer vision algorithm built in that helps you identify stuff from photos as well. Super cool. But our thought is that if we could get just a small fraction of the people, the visitors in the park using iNaturalist, we'd have thousands and thousands of new records and thousands of eyes around the park seeing things that we can't necessarily see. By the way, we have a staff of 3. You have 33% of Discover Life in America's staff here right now. So we have been kind of, so we call this the effort to get people to use iNaturalist Smoky's Most Wanted. And we have really been like pushing this for the last 3 years, and we are up to over 150,000 observations in iNaturalist, and those are of over 6,000 species. That's about 28% of the known biodiversity in the park, which is really cool. In fact, almost 200 of those species were new park records. We were able to verify them. They're specimens or species that we're able to identify from photos, which can't do everything, or we are able to verify in some other way, and they're new to the park, so that is really, really cool.
The iNaturalist comes with a GPS location, right?
Yeah, it does come with a GPS. So when you use the app on your phone, it just uses your phone's GPS. You can also take photos with like a digital camera, upload them via their web interface, and add a location as well. Yeah, so there are all these georeferenced data points representing tons of different species in the park. And so we can use these data for creating, to model species distributions across the park, to look for exotic species. So we are on the lookout for the spotted lanternfly, which is not in the park yet, but has been seen in Tennessee. I think just Tennessee. I don't think North Carolina yet, but I'm not totally sure about that. But it's a matter of time, and so the more people we have out looking for it, the better that we can maybe control it beforehand. So I mentioned that there are a ton of different people on iNaturalist that will help us identify things, and we get some really great expert level identifications. So one of the things that we've been doing is, apart from sending material out to experts, is to just photograph it, get some decent diagnostic photos that work for a lot of different taxa, and post them to iNaturalist. And there are people from all over the world that will identify stuff. This is a new species record on the right of a dryaenid pincher wasp that was photographed by one of our volunteers that has this really great focus stacking setup. And it was identified by a person in the Netherlands who happens to be an expert on dryaenid wasps. And it's a new species record, new genus record for the park through this community science platform. So that is super freaking cool. All of these are new records for the park as well that we found. This one I just nabbed randomly while giving a wildflower hike to some folks, and it just happened to be a new species record for the park. It really is not that hard. There's just so much biodiversity there. So just a few to finish out with a few stories of other discoveries. So last November, we held the first of what we're kind of made a series of these volunteer-led blitzes. Actually, I should say the first since the early 2000s. We haven't done this in a while. Anyway, we went out to Cades Cove, which is this open meadow on the western side of the park. We had just a few volunteers with us, and we just went out and looked for all of the galls and leaf miners that we could find. So galls are these cool structures formed by different insects on plants, and then leaf miners are these little trails left by, or leaf mines or trails left by different insects mining through the top and bottom layer of leaves. And so we had it was about 25 person hours, and we made 156 iNaturalist observations, and we found 8 new species records, new taxon records for the Smokies in just like 5 hours. Really, really cool, very effective, and literally this is the first one that I've ever led, and we found so much stuff. So we're definitely doing this again next fall. We're also working with Andrew Forbes' lab at University of Iowa to send them a bunch of gall specimens to identify. Charlie Eisman, who's possibly like the world expert on, or one of the leading experts on leaf mines and leaf mining insects, is on iNaturalist and is constantly checking and identifying stuff. So leaf mines and galls are actually really fun on iNaturalist because they seem like they would be like impossible to ID, but there's a handful of people that will very quickly identify stuff for you, and so you get that like rush of like learning and discovering, and yeah, super cool. Just to give you an idea of some of the other volunteer-led activities that we have coming up this year, and I think next week we're doing a pollinator survey for some of the early wildflowers in the park, mosses and bryophytes, some more pollinator stuff in the high elevation. We're actually going to do some surveying for fireflies in Cades Cove, lots of other things. Myco Blitz that we participated in last year, monarch tagging. There's monarchs that migrate through the park that we tag. All kinds of fun stuff. So another little discovery story. We have 19 firefly species known to the Smokies. A lot of people come down there to see this one, the synchronous firefly, and also my favorite, this one, the blue ghost firefly, that comes out at the same time. But a couple of those are in this complicated group, the Photurus. So Photurus are really difficult to identify. You really need to have like both morphology and be able to see the flash patterns, and the flash patterns can vary, and it's just a whole mess. So we, just as we were initially starting to barcode things, I barcoded a Photurus specimen that I collected last May, and we were able to barcode it. It amplified correctly, sequenced correctly, and it matched with a Photurus quadrifulgens in GenBank, and I don't always trust the IDs on some of these species, especially like hard to identify species. But in this case, this was one that was identified by one of the like experts in probably the biggest expert in the U.S. on this group, the Photurus. And so we feel pretty comfortable with it. So this is a new firefly record for the park, yeah, so that's really cool. We don't do that very often. So now we're hooked, and all the specimens in blue we have barcodes for. I guess all 3 specimens, but all the ones that are in bold we have represented in the Natural History collection, and so we're going to start barcoding those. and put names or put verified names on a few of them, but also contribute barcodes to the database. And I think this is my last little story, is we've just started this concept called an ID Blitz, where we just bring in a bunch of taxonomic specialists that work on a particular group of organisms. In this, in our inaugural one, we started with flies, and just spend a few days blitzing through identifying material that we already have, like that ATBI backlog material, and also the material held in our natural history collections. And so, in just 3 days, this group of 7 people identified something like 500 flies beyond family level, and there are probably 49 different taxon records for the park. They also found in the literature some species, 6 species that were described from the park that we just didn't have on our fly list yet for whatever reason. And so we have added more than 50 species records just in 3 days, and it's really cool and just really fun to hang out with these folks. So we're super excited about doing more of these. We'll probably do micro Hymenoptera next, and probably another fly one, maybe some Hemiptera. We'll see. And last little thing, I just want to plug our summer internship. It is closed for this year. We've already selected our interns, but next year it opens, the application opens January of every year. And you basically just get to run around the Smokies with me, learning about the flora and fauna there, learning how to do DNA barcoding, doing field research. You get to work in a national park, which in itself is super cool, but you get to work with national park staff, and it does come with a stipend and housing, and it's a 10-week thing over the summer. So I highly encourage anyone interested, especially undergrads, although we do sometimes take grad students as well. You can find out more about it on our website. And so all of the slides are located at this QR code here, if you want to go back and take a look at anything. And I would be happy to take any questions. Thanks for having me.
So with a backlog of 8,000 specimens, have you thought about flipping things and barcoding first? And that can get you to, family or multiplying out most of your genera probably pretty quickly. So next gen now, $1,600 you can do 300, 400, 500, 600 single lineage, and actually speed things up and target your morphological.
Yeah. It's been brought up.
The ethanol that these things are sitting in, we do it all the time. You don't even have to rip it up, and especially if they've been in the ethanol for a couple of years.
Yeah. Ooh, we should talk more about this.
You'll be talking later.
Everyone in it.
Perfect. Excellent. Yeah.
Actually, I have a follow-up. Your
Yeah.
collection, maybe you said at the beginning, I apologize, but where does your collection infrastructure funding come from? Is it-
Yeah, I was gonna ask that.
Yeah, good question. So the collection is actually national park. So it's we contribute material to it, but it is owned by the national park. So all the infrastructure and all the funding comes from the National Park Service. It is underfunded, I will say. There's currently one staff member that works over this collection and another one that houses archaeology and records and things, cultural things as well. But he's about to hire 2 new staff members, so at least it'll be a little less bad. But
I'm noticing you don't have like NSF or any other little logo in there.
no, we this is mostly funded. Our funding comes mostly from private donations and fundraising events and things like that. Yeah.
I mean, it's a fantastic teaching tool, so NSF
Yeah.
technically.
Yes, definitely. I think back in the day, actually, me back up. Several ATBI related research, several researchers contributing to this project have gotten NSF grants. We just haven't as an organization, but it's definitely something on the horizon.
I have a different question.
Yeah.
Because you showed in the beginning a lot of the diversity. I also, I work for Dan Jansen, so I know the big picture thing that came out of his head. And so you didn't really talk about the species interactions at all. Okay. But the other part of it, maybe I missed it. How many of the species are endemic to the Smokies?
I didn't say. I think it's somewhere around 200, but it actually might be more than that. Across the board.
Okay. It'd be kind of interesting to provide like how different is it? I mean, you just said it has an enormous species diversity in trees and all sorts of things.
But how different is that from
it's
just
quite
outside the park?
unique.
Yeah, we have. Let me revise that to several hundred endemic species. I can't remember the number off the top of my head. And a lot of those are just high elevation species, species that have, so the Smokies was never glaciated, and so there's sort of like a refuge for a lot of species.
It's been there for 500 million years, so
It's been there for a
some
long time,
Right.
yeah. And there are a lot of species that are found here and then Canada,
Right.
that are like refuge populations, which is really cool as well. Yeah. What else?
So I gotta ask, do you get a lot of retiree volunteers to help you?
We do some, yeah.
Okay.
Yeah, we do. There's a ton of there are a ton of volunteers that work specifically in the Smokies, like that are volunteers for the national park, I should say. They have like 15 or 1,800 volunteers, including one that works in the collection. Tracy, I don't know her last name. Story. Thank you. But there's actually this fun group of folks that are retirees mostly from the Park Service. One of them is retired, like the father of this program that really pushed to start an ATBI in the Smokies, Keith Langdon, and he and his band of interested naturalists are called the Grizzleds, the gang of retirees in search of life's diversity, I think, is Grizzled. It's a great acronym too, and they're sort of like the rock stars of the ATBI. They just go to all these different to rare habitats, interesting seeps, places that just haven't really been visited very much, and blitz everything they can find there. There's different members of the group are really good at like lichens. One of them's really good at bees and flies. Like they each have their different interests, and so they've contributed thousands of new records for the park. Yes.
So what is a blitz?
Yeah, good question. I keep using this so it's the original concept was it's called a BioBlitz, and it is like a focused effort to catalog either everything in a particular area for usually kind of a short amount of time, but a lot of times they're very taxon focused. So like our gall and leaf miner thing, we're specifically looking for a couple, but it's like focusing on a particular taxonomic group or life in general, but like in a usually with bounded in some area. Like here, there is a Rutgers-wide blitz going on via iNaturalist.
Yeah, I was gonna plug it. Join the Rutgers Personal Bio Blitz 2024. Yahel and I are in the top 30.
Nice.
Oh, you're tag teaming?
No, he's like 12 and I'm like 20. But
I was gonna say, if you're both working at throw that in there.
well, let's say come beat us.
And so that is, as I understand it,
It's an
that's
iNaturalist project.
yeah. So
Yes,
you
project
you
proposal?
contribute stuff through iNaturalist anywhere around campus.
Any Rutgers person can do it anywhere.
Okay, cool. And it's till May or something?
Rutgers Personal Bio Blitz.
Yeah,
It's
that's
until May something.
cool. Very nice. Yeah.
Yeah,
Anybody wants to
it's
come
Lead
to
Us
the museum
Through
and
Ways
gets
project.
I'll unlock the door for you.
Well, it has been something collected between March 2nd and May 15th
Yeah.
or something like that.
That's a good idea.
I love it.
Actually.
Yeah.
Well, that would be-
So I have a
Oh,
follow-up question.
Sure.
Because we also did have here cichlets, and you mentioned mites.
Mm-hmm.
And you also mentioned species interactions. So how
Mhm.
much has there been in terms of parasites? I mean, we talked about parasitoids, but you looked at ticks.
Yeah, so there are, the ticks are included in that, the big mite count. Yeah, there has been quite a lot in the ways, looking at ticks, also like bird mites and or feather mites, and looking at the interactions between the different, they're mostly kind of species specific to different birds, but yeah, cataloging those.
People doing mammals, are they looking at ticks in mammals?
Yes,
Yeah,
ticks, and also we've gotten like a handful of viruses from bear viruses and things. Yeah, so there definitely are some.
This is like specific groups that are doing that?
It's mostly specific researchers that are focusing on one particular taxon, or that are really interested in this group of parasites or parasitoids. Yeah, a lot of that is kind of like researcher led by their own group of interest rather than like our kind of blanket approach. But parasites are high up on our priorities.
They are? Okay.
Yeah, especially those parasitoids. Yes, sir.
Just building up the question Dr. Fonseca had asked earlier about like species that are endemic to the Great Smoky Mountains. How, like, so from the time that like the initiative has started, how do you determine whether or not something was actually like a species is actually like native to that area as opposed to recently moved in like shortly before like the initiative started or something like that?
Yeah, that's a good question. So, a couple of things. Most of the species, I would say most of the species that we're finding have been in the park the whole time. A few of them have moved in. There's definitely a few exotics. A few things like armadillos are now in Great Smoky Mountains National Park, are a recent addition, and they have just been slowly expanding their range naturally. But yeah, to really determine somebody like a species' range, there really has to be like more. We have to have a lot of data points. There has to be more. Usually, it's like a taxonomist who will. Let me back up. A lot of those are like lichens. There's actually some folks from the New York Botanical Gardens that come down and work in the Smokies a lot, and so they know their lichens from all over the eastern U.S. and because they've studied them in all of these places, they can tell, oh, this is something that has a very small range. We've sampled consistently across, and can tell. But yeah, if it's just like something that we find one specimen of, we have no idea. It takes some digging into.
Thank you.
Mm-hmm.
So you mentioned climate change
Mhm.
having an impact. You have this massive data set. It doesn't go back very far though. So are you at the point yet where you can start measuring changes in species diversity or abundance with your collection?
Yeah, that's a really good question. It's something that's been coming up a lot. We've thought a lot about like insect decline, being able to tell differences in abundances and diversity at these different sites. And we haven't, in hindsight, I wish we had done some of the sampling a little bit differently. If I could go back 26 years and change a few things. There probably are some ways. Actually, there's a study out of one of the UCs, UCLA, I think, of somebody who is going back to a bunch of sites that were where high elevation sites where insects were cataloged in 1974-ish, went back a few years ago and tried to resample all of those sites in the same way, and is comparing the results. I think he's involving birds with that as well, but anyway, yeah, so to answer your question, there's probably some ways that we can look at things like insect decline and changes in species diversity over time. For some taxa, better than others. Oh, we have like 30 or 40 years of aquatic benthic macroinvertebrate samples from all across the park. Yes, that Liz has contributed. So for specific groups like that, yeah, we definitely can look at changes. Fish, there's been a ton of fish monitoring. Fish. There's been a lot of misnetting, so birds, we have some pretty good, pretty consistent long-term data sets. So yeah, so for some of these taxa, we have...
For the fireflies, you guys do a lot of outreach for the fireflies.
We do a lot of outreach for the fireflies. As far as consistent data goes, I don't know how far back that goes. That really is kind of blown up since the 2010s, maybe. Yeah, so the synchronous fireflies draw tons of people into the park. There's actually, during their peak flashing season, which is like first couple of weeks of June usually, it's gotten to be such a big thing that the main area where people go to see them is shut down, and there's a lottery system to get in. We, as a nonprofit organization, have a fundraising event at the same time at some private land just north of the park, and so we have these small groups of people that we show the fireflies, and it's a really cool thing. You have to pay for it, but it's funding biodiversity work.
You don't see any pattern, because that's one of the groups that people are arguing are declining is fireflies. Do you see any?
In the park, it doesn't really seem, they definitely vary from year to year. Like there was some definite effects from drought, big drought a few summers ago. That was like a couple years later, we had a big drop in the firefly populations, the synchronous firefly population specifically, but yeah, in the park, they're actually doing reasonably well. Outside of the park, not as much. There's a ton of development in our area and land use change and such that is definitely affecting them. But in the park, they're okay so
Well,
far.
but that's an important point, is that outside of the park, you are seeing a decline.
Yeah.
Well, that's kind of the point, right? I mean, if you keep
Yeah.
things under control, they
Yeah.
don't go down. If you have encroachment, they do.
Yeah, that's a very good point.
I
Yeah.
mean,
This is working.
Sorry, do you have, is this acting as like a biological station where you can, there's like facilities, like places where students can stay, faculty can stay? Is that how this is working?
We would like to be more like that. There is one field station in the park and two field stations nearby. One they're owned by different universities. But it would be great if we had like for this fly ID
Like a
blitz.
dorm?
It would be great if we had some kind of dorm or something like that. Working with the National Park Service, there is so much red tape involved with anything, especially apparently housing, which is a huge problem in the area. Anyway, it's a lot. Yes, ideally yes. Not at the moment, except for in limited circumstances. But the field station that is in the park called Purchase Knob, or the Appalachian Highland Science Learning Research Center, or something like that. Anyway, it's in the southwest corner of the park, and it is really beautiful and very close to a bunch of neat sites, and that's where all the lichen
it's
folks.
associated with the university.
That one's actually associated with the park, so any researchers can come in, and it's like there's like a nominal fee of $10 a day or something like that, and it's got dorms. It's got so I'm happy to connect anyone who is interested with the park folks to.
This is a shout out for biological stations. I mean, biological stations have been on the decline over the years, and
Yeah.
it's so important.
They're extremely important, just to be able to be so close and to literally go out your door and
Yep.
sample all the stuff that's there. Liz had a oh yeah.
It wasn't quite a question. I just wanted to add.
Yeah,
I remember last year, Becky had mentioned that the fireflies were emerging a lot sooner than she had expected because the way that they calculate when the emergencies are happening using
Degree days.
Yeah. Where there's one in the ground, there's one in the air. She takes the little pegs out, puts them in a computer. That's all I know. But
temperature
yeah,
temperature.
So she's like, "Oh, Liz, do you think you can find them this time?" I'm like, "Sure." She's like, "Warmer, colder, 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 colder." I'm like, I have no idea. Show me where it is. But yeah, so actually last year, I think it happened like the end of May rather than the beginning of June. Just like a climate change thing. But like,
When I went down there, we got the lottery, but I think we were supposed to be 5 days off of the peak, and we managed to get there the day. I think you said it was going to be predicted to be the peak. Well, took us to the day. It was phenomenal.
yeah, I was working on peak day last year. It was great.
This was a. This was a while ago.
2019-ish?
I also wanted to ask, I didn't know your whole like backstory of everything. I knew you were in New Jersey at one point and don't like Corporal, that's it. Were you working, she mentioned you were working with dragonflies in it. Were you involved with the mercury stuff?
No, I did. So there's effort to sorry.
No, you're okay.
There is somebody's working on a study looking at mercury bioaccumulation in dragonfly nymphs in the streams. I've helped with that, but I was not originally associated with that or anything.
You know?
I just know dragonflies.
Got given to Becky. I don't remember who it was originally.
Yeah.
That's why I'm like, wait, was Will doing that this whole time?
Yeah, also dragonfly related, but different thing.
All right, we do have, I think, one extra spot in Will's schedule if anybody wants to talk to me and sign up for later today. But otherwise, thank you, Will. And the students, we're going to go next door.
Oh, forgot to ask.
Oh, whoops.
Sorry. Sorry, folks on Zoom.
I already turned that off.
Oh, okay.
You're on mute, so.
I just like walk around and take pictures of random stuff. I put a
Hi, I don't know what you meant by like a sign up spot or whatever. Am I allowed to join you today in the classroom?
Alex, did you do that today?
Oh my god, that was so good!
And so, how do you sign up?
What were the new EPTs? Now I'm like going through all of them in my head. I'm like, which one? What were they? Do you know?
What were the? I have a list somewhere in my email. But,
Becky never told me this.
No, that's just.
No, it's just like
You good? Oh.
climate change. Yeah, we addressed that.
Oh, sorry.
No worries.
I forgot about that, so. Okay, thanks. I'm just gonna exit. Do you need a break for a second before we go have pizza?